All right, so this recording is for you to work on your pear deck asynchronously. If you missed the class time when we were going over and going through these slides. So the do now that you're going to answer on the slide is using one complete sentence. What surrounds the outside of a cell that controls what goes into and out of a cell? Our learning objective is that you will be able to describe the structures of the semi-permeable cell membrane and identify examples of cell transport. So we're going to talk about the thing on the outside of the cell today, and we're going to talk about what it does for us. So before we get into that, we need to understand what homeostasis is. Homeostasis is the process by which organisms keep their internal conditions relatively constant despite changes in external environments. So keeping the inside constant when the outside changes. Think about things that your body does that keeps constant so that you can survive, even though the outside is constantly changing, whether it's the weather or the season or just the temperature of the day. So examples are like sweating. Let me explain. When it's too hot outside, your body decides that, hey, the temperature is kind of too high in here. And your body processes need a certain range for them to work. So in order to keep that temperature within a reasonable range, your body is going to produce sweat. And as the sweat evaporates from your skin, it takes some of the heat energy away. And as the heat energy leaves, your body is therefore cooled down so it can maintain a specific temperature range in order for you to survive and function. Otherwise, if you got too hot, you would get heat stroke and you could die from that. I want you to try to explain shivering now. How is shivering an example of a homeostasis? I'm going to ask you to type your response out. The osmotic regulation one is an example that we're going to talk about in the next slide set that we go through. And it's related to individual cells in your body and how they regulate the amount of water inside and outside of them so that they can maintain a certain amount of salinity or saltiness or sugariness so that they can function properly. So again, on this slide, explain shivering. How is shivering an example of homeostasis? How is it an example of maintaining an inside condition while the outside might be changing? So the cell membrane, this is a picture of it. It's called the phospholipid bilayer, which means there's two layers of lipids. Bi means two, bilayer, two layers. Phospholipid is the component of which there are two layers. And the cell membrane, as you can see right here, there's one layer across the top and then another sheet on the bottom. And if you notice, the circle and the wiggly tails are actually facing each other in this bilayer. It's called the fluid mosaic model because it's kind of basically like a liquid surface. If you ever looked at the surface of water, that's what the cell surface almost looks like. Okay, it flows just like the surface of a liquid, therefore it's called a fluid. And it's called a mosaic because it's made up of, as you can see in this image, a bunch of different things together, like a mosaic you would do using glass or using tiles to create an image. And lastly, it's selectively permeable. Now, this term is super important because selectively permeable means that permeable means that things can go through. And it's important for us that things can go through the cell membrane because we've got to exchange materials between cells. However, it's selectively permeable, which means that only certain things can go through, because if everything can go in and out of your cells, then your cells would either explode or random things would be in there that shouldn't be in there, or they wouldn't be able to get rid of things that they need to get rid of. So it's selectively permeable because it has specifications on what can go in and out of the cell. So the cell membrane is actually in charge of maintaining homeostasis in the cell. So every single cell is surrounded by a cell membrane, which was the answer to your do now. And it's important for the cells to have a cell membrane because it's not the nucleus that actually controls it. The cell membrane is actually more of a brain than the nucleus is because it controls everything that goes inside and outside of a cell. And that helps it maintain homeostasis. So maintain the inside of a cell, even though the outside of a cell, the environment might be changing. So this is a phospholipid. So this is the thing that we talked about earlier when we said phospholipid bilayer. And every single phospholipid has a phosphate head at the top. There is a glycerol that connects that phosphate head to two fatty acid tails. That's why when you look at it drawn, it's always a circle with two little tails. It's one phosphate head and two fatty acid tails. So the head is polar and the tail is nonpolar. So the head likes water and the tail doesn't like water. Okay, phospholipids, when they form a single layer, this is kind of what they do. This is kind of what they do inside of soap. They help you wash away things like oil off of your plates and your dishes. This is what they do. But when they form a bilayer, that's the cell membrane. So there's going to be water on either side. And the head of the phospholipid likes water, but the tails really, really hate them because they're fatty acids. They're like fats, right? They don't like water. They don't mix. So they actually stay together, but the phospholipid heads like the water. So they'll touch the water. So they form this bilayer where the tails face each other and the heads face outward. So you're going to use your drawing tool and draw text boxes to fill in as I talk. So the phospholipid structure is, the head is a polar phosphate group. That's polar phosphate group. The middle is a glycerol, Glycerol. The middle is a glycerol. The two tails are fatty acids. The two tails are fatty acids. Now polarity. Hydrophobic means, well, hydro means water, and phobic is when you have a phobia. So hydrophobic would mean water fearing or afraid of water. On the other hand, again, hydro means water, right? Philic is the opposite of phobic. So the opposite of completely fearing something would be probably loving it, right? So hydrophilic means water loving. And that would be hydrophilic or loving water. Now, my examples are actually wrong. So I'm going to ask you, if you catch this like I did, to use your drawing tool and cross out the examples. They're actually the opposite. So I'm going to be checking this slide to make sure that I see that you crossed out the examples and swapped them, because we know that the phosphate head actually likes water. So that should be hydrophilic. And the fatty acid tails hate water. So they should be hydrophobic. So make sure you got that cleared up on this slide as you filled it out. If you need to, you can always just replay sections to fill out. So other important molecules in the cell membrane are transmembrane or integral proteins. They provide channels for facilitated diffusion. We'll get into what that is in a second. So when you're moving molecules through the cell membrane, sometimes you need a protein channel just for helping. There's peripheral proteins that help facilitated diffusion work faster by recognition. They're just kind of like signaling markers like, hey, you're going to come over here and you're going to go through the cell membrane here to certain molecules. And then there's cholesterol, which actually, when we think about it, cholesterol is bad if it's in your heart, but inside your cell membrane, it's actually super, super important, because actually, if you didn't have cholesterol, your cell membranes would break and rupture all the time. So they actually help the phospholipids stay connected and stay sturdy as a cell membrane. So this is what they look like inside of the cell membrane, as we see our phospholipid bilayer across the middle right here. And we have peripheral proteins that are only on one side. We have integral proteins, which are completely through as a channel through the membrane. Then we have cholesterol that are these tiny little 4-ringed molecules that are randomly inside of the cell membrane at random points that help keep it sturdy. And as you can see here, there's a phosphate head that's polar or hydrophilic, and there's a fatty acid tail that is nonpolar or hydrophobic. So we have selective permeability, like I mentioned before. Cell membranes are selectively permeable, which means only certain things can pass through. Certain things, selective. So things that can passively cross the membrane without using energy are nonpolar molecules, small molecules. Things that need actively passing through or need energy to pass through the membrane are things like polar molecules or things like large molecules, just because if you try to shove a large molecule through, it'll rupture the membrane. Or if it's a polar molecule, only the polar heads of the phospholipids like it. The fatty acid tails really hate it. So you're going to need something to help it get through that fatty acid section in the middle of the membrane. So I'm going to ask that you select the correct answer. Because the cell membrane is selectively permeable or semi-permeable, it will allow certain types of molecules through, but not all. The types of molecule that can pass through the cell membrane the easiest are the ones that are both. And I'm going to ask you select your answer. Select A, B, C, or D. The answer is C. So molecules that are small and nonpolar move through the cell membrane the easiest because they interact with the fatty acid tails and they like each other and they're small, so they're not going to rip the membrane. So there are two types of cell transport, and I keep mentioning it, but we're going to actually talk about it now. So there's active and there's passive. And the first one we're going to talk about is going to be passive transport. So passive transport, as you can see, is transport that requires no energy. So we're not going to be using up any energy. It just happens naturally on its own. You don't got to do anything, and it just happens. So we always are going to move along a concentration gradient in passive transport. So a concentration gradient is a difference in the concentration of a dissolved substance in a solution. So there's a high side and a low side. There's something that's like really, really salty, and there's going to be a side that's not that salty. Or you can think about sugar. There's a side that's really, really sugary, and there's a side that's not that sugary. And so that's a gradient, because from one side to the other, you can move from high to low or low to high. Passive transport is always going to be movement of materials that does not require energy, right? Energy usually comes in ATP. We're not using it here, okay? And in passive transport, we're always moving from a high concentration to a low concentration because it just flows easily that way. Reason is because passive transport is like rolling down a hill. If you're riding a bicycle down a hill, you really don't got to put into any energy into it, and you'll still go. You're going to go from a high concentration at the peak of the hill to a low concentration at the bottom of the hill in passive transport, and you don't have to put any energy into it, like rolling a bike down a hill. So what is happening here? Well, this is an example of passive transport. So you're going to write your response in your own words. Okay, I'm going to ask that you describe what's happening. If you drop ink into a beaker of water, what's going to happen to the ink molecules after a little bit of time? Well, the answer is that they're going to spread out because initially there's a high concentration in the middle and a low concentration in the surrounding water. But after a long time, they're going to move from the high to the low. So they're going to move out into the open space where there's low concentration, and they're going to be evenly spread out after some time. So that's actually what's happening, and that's just an example of passive transport. So this is actually diffusion. Diffusion is a type of passive transport. The passive movement of material from an area of high to low concentration is diffusion. So any molecules moving from a high concentration to a low concentration and spreading out evenly is going to be diffusion. So again, a high concentration to a low concentration in comparison is if you have the same amount of stuff, but you have a lot more molecules in that same amount of area, that would be high. And if you have less molecules, it would be low. And if you take them and put them together with a semi-permeable membrane, you're actually going to have these molecules diffuse across the membrane towards the low concentration until they are evenly distributed across the entire space that they're connected by. As you can see here, here's an example over here. Originally, we have a lot more concentration of the red molecules on the left side, which is sugar, and we have even amounts pretty much of the water molecules, but a little bit higher concentration on the right side. And after diffusion happens across the semi-permeable membrane, notice that it's labeled over here, you're going to actually have an even distribution or an even amount of the sugar on both sides and of water on both sides. So concentrations will be equal now, right? There's no high and low anymore. The before over here had high and low. Over here, there's no high and low anymore. The example that we can talk about is also like farts, right? It's like the ink one. In the beginning, if somebody farts in the back of the classroom, I mean, it's concentrated back there, but the rest of the room has a low concentration of fart molecules. Through the process of diffusion, the fart molecules will slowly spread across the room, and this is the same number over here and across the room. And we're going to end up with a uniform concentration of fart molecules across the whole room. So at that point, everybody smells it. Or if it's spread out enough and it's not that bad smelling, nobody will smell it. So again, diffusion overview is diffusion is the movement of molecules from an area of high to low concentration, and it is a type of passive transport, meaning that it requires no energy or no ATP. Now, osmosis is specifically the diffusion of water. So it's the movement of water molecules, and we'll talk about this in a complete slide on its own. But water is essential for life, so it moves across membranes easily because it's a small molecule, even though it's polar. And again, this is passive transport. So we're moving from high concentration to low concentration, but there's no ATP required. So if you look here, we're actually, the semi-permeable membrane is actually not going to let these circular molecules move through, but it'll let water move through. And in order to equalize the concentration on both sides, because these molecules can't move, the water will move from the left side to the right side. And in the end, you'll actually get same concentration, but one side of the beaker will actually have a higher level of liquid than the other, because that's how the water is going to move in order to equalize the concentration. So there's no high or low. And there are three kinds of solutions when you're talking about osmosis. It's going to be hypertonic, isotonic, and hypotonic. And we'll get into more details in the next class. Facilitated diffusion is diffusion of a larger or polar molecules through a protein channel. Because if you're moving large molecules or polar molecules, the cell membrane doesn't like that. The cell membrane could rip. So we're going to use special protein channels, which are basically acting like car tunnels, right, when you're driving, to move the stuff that we're trying to move through the cell membrane. And these protein channels are more sturdy than membranes, so it makes it a little safer to move them. And again, it's still passive. It's still high to low, no ATP required. And the last thing we're going to touch on today is the other type of transport, which is active transport. And that requires energy. It requires the usage of ATP. And so we talk about active transport, like trying to ride a bike up a hill. If you're starting from a low concentration and you're trying to push something up to a high concentration, you're going to have to put energy into it. It's like riding the bike up the hill. You're going to have to pedal pretty hard and use up some of your energy to move from the low point to the high point. So there's some examples like endocytosis, which means you're going to move things into the cell. Endo, into, and that's using ATP because sometimes things just aren't supposed to be going into the cell very easily, but it's like eating, right? Cells got to eat, cells got to drink. That takes energy, right? You got to swallow the liquid, swallow the food. The cells are kind of doing a similar thing. So we have phagocytosis as a picture on the left side, which is when cells are eating solid things. So they'll reach out with their two little sections of their cell membrane and they'll grab things and bring them in. And that takes energy because they're actively moving to do it. On the right side, we have penocytosis, which is the intake of liquid. So if liquid molecules are around, cells will actually let them come into the cell membrane and engulf them inwards. And again, that requires energy because cells are moving to do this. So endocytosis is moving stuff in using energy. Here is a video. I'm going to ask that you describe what is happening in one complete sentence. So this big blob on the outside is an amoeba. And the little thing is a little paramecium or a single-celled organism that the amoeba is going to be doing something to. And what it looks like is that the amoeba is wrapping these two tentacles around this thing. And slowly it's going to be getting blurrier and blurrier because it's injecting digestive enzymes. And eventually it's going to be doing something related to the previous slide that we talked about here, where that little thing is getting eaten, but you have to tell me whether it's solid or liquid and if energy is being used or not. Okay, exocytosis. Exo is using ATP to move things out of the cell. Exo out, exo out. Okay, it's for removing waste and exporting protein products. So when we do the central dogma and you make proteins, you got to move them to where they got to be used. Right now, all of the cells that make the proteins use the proteins. So sometimes also cells got to basically poop. Right, cells are going to eat, like phagocytosis. They're going to have to poop or get rid of waste, and they do this through exocytosis. It takes energy, right? If you're pooping, you got to push. Cells got to push things out of their cells too using the cell membrane, and that takes energy. Sometimes they'll use protein pumps, which will help pump things out of the cell, and that takes energy because it's literally a pumping station, right? So here is a sodium-potassium pump. Describe what is going in and what is going out in one sentence.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Okay, so that was the sodium potassium pump. If you need to rewatch sections of it in order to describe for me what is going into the cell and what is going out of the cell, you can do so. And remember that this is using energy because it is a pump. So this is an example of active transport. Okay. So I'm pretty sure that is the last slide. So once you finish this, make sure all your Pear Deck slides are completed for the ones that I asked you to write responses or select responses for. And then you're going to go ahead and work on your interactive notebook section, should be section 1. And then there's an exit ticket that's a couple questions. If you have any questions on your Pear Deck, you can always go ahead and rewatch sections of this video because I will be checking the Pear Deck for completion. So you need to make sure that it is done.